So I certainly do want to welcome everyone to today's webinar. This is indeed our, I believe, 16th webinar now. And actually, let me go back to the beginning real quick. So again, we certainly do welcome everyone. So before we begin the session, I do want to just share some general news on behalf of the EMN. So my name is Johannes Fairman here from MD Anderson University here in Texas, and I will be hosting this webinar. So as a general reminder, we do encourage all participants to connect with us online through social media. We do have a variety which are demonstrated here through Facebook, Twitter, etc. We do also encourage members and anyone really new to the field to take advantage of our Metabolomics forum. So if you do have any questions that relate to metabolomics, please do feel free to go to this forum and ask questions, and we can certainly provide answers or at least give you some general insight as to whom you may contact to derive answers for your respective question. I realize it's still a little bit of a ways, but I do want to encourage and remind everyone that we will be having the next Metabolomics conference in Seattle, Washington on June 24th through the 28th. So do keep this in mind, and certainly if you get an opportunity, we encourage everyone to participate. This is certainly a great venue to really get more immersed in the field of metabolomics. So with that, we will go ahead and begin our webinar. It's really a great honor for me to present today's webinar speaker, Dr. Lane. He is certainly a very experienced individual as it relates to stable isotope resolved metabolomics, and I think really a good pioneer of this sort of technology with over 30 years of experience. So Dr. Lane is currently the co-director of the Central Environment Systems Biochemistry, which is responsible for developing and applying new methods for metabolic studies. Dr. Lane's background is in biochemistry, enzyme kinetics, and NMR, and particularly appropriate for designing stable isotope resolved experiments and their execution and interpretation from a biological standpoint. Furthermore, Dr. Lane does have plentiful experience in computational modeling of enzyme and NMR dynamics, and alongside Dr. Teresa Fan and Dr. Richard Hagassi at the CESBB, Dr. Lane has been developing stable isotope tracer approaches really for studying cancer metabolism. It is really a great honor for us to have Dr. Lane here today, and it is a very exciting topic that is dear to my heart, so I certainly always appreciate listening to this sort of talk. Now, I do encourage that all attendees please do remain muted for the course of the talk. If you do have any questions, please use the questions tab. We will go through these at the end of the webinar, for which I will be moderating. But without further ado, I will pass this over to Dr. Lane, and I'm sure that everyone here is in for quite a treat.
Okay, good morning and or afternoon, everyone, and thank you, Johannes, for that very nice introduction. So the first thing is, can everyone see my screen? Looks like probably yes. Okay, then I am going to begin in the absence of any other feedback. So as Johannes said, my name's Andrew Lane. I'm one of the directors of the Center for Environmental and Systems Biochemistry at the University of Kentucky. And this is a quasi-independent unit at the university in the College of Medicine. And it is funded in part by one of the Common Fund's metabolomics centers. And our speciality and focus is the use of stable isotope resolved metabolomics, and our particular research is cancer metabolism. So although we do a lot of collaborations with people throughout North America and elsewhere in the world, most of our work is cancer related, but we do work with people who are interested in cardiovascular problems or other diseases. But the examples I'm going to give are from our own work, mainly because I've got the data available, which will be cancer oriented. So with that, how do I go forward? There we go. So I'm going to spend a brief amount of time on explaining what stable isotope resolved metabolomics is and why you would want to do it, which is probably more important. So a little bit about the basic approach to things you have to think about in terms of experimental design and what kinds of approaches you would use to incorporate stable isotope traces, and then go on to what model systems can be this can be applied to, and then the primary outcomes of this kind of approach is reconstruction of metabolic pathways inside cells, inside tissues, that give you the mechanistic information about the progression of or response of the disease to intervention. And I'm going to say not much about the two major technologies that are used, because I think most people are aware that mass spectrometry in its multiple guises, whether it includes one of the many kinds of chromatography prior to the ionization and detection in the mass spectrometer, and also NMR, and we use both of these technologies in parallel. And then finally, mainly because it's often stated in public or even in the literature that stable isotope resolved metabolomics automatically means flux analysis. This is not correct. Flux analysis is its own thing that usually needs stable isotope traces, or even non-stable radioactive traces can also be used, but you're looking at the rates of reactions, of particular biochemical reactions, either inside the cell or for the uptake and release of waste products from cells in tissues. And that's requires that you know what the pathways are, but it's an additional step after you've done all of that work. So I will, if I have the time, say a little bit about that at the end of this presentation. So stable isotope resolved metabolomics, or SIRM as we now call it, what is it? So everyone knows about metabolic profiling, where you measure the level of metabolites, their total amounts in biofluids such as plasma, serum, urine, cerebrospinal fluid, and so on and so forth, or in tissues if you get access to needle biopsy or punch biopsy or tissue discarded from surgery. Generally speaking, because most metabolic pathways are embedded in complex interacting networks, it's very difficult to determine where each atom that goes into a particular metabolite came from. So profiling, especially of biofluids, doesn't really give you mechanism in general. So typically it's used for biomarker discovery, and what you can do with biomarkers obviously is look at progression of a disease, response to intervention, and so forth. The stable isotope resolved metabolomics, in contrast, uses unsurprisingly stable isotope traces, although traditionally one could use radioactive traces for the same purpose, and that it requires that you introduce into the system, whether it's cell culture or tissue or an organism, including human patients, a stable isotope enriched precursor, such as C13 enriched glucose or glutamate or whatever your favorite thing is, and then as a function of time follow the fate of each atom, each carbon-13 or nitrogen-15 or whatever the enriched atom is, through the metabolic pathways as it's transformed from the precursor molecule to glucose through, for example, glycolysis ending up at pyruvate or lactate and entering the Krebs cycle and so forth. And that generates what we call atomic fate maps. So that tells you exactly which atom in which molecule that you're monitoring, where it came from. Did it come from glucose directly, or has it gone through several other transformation pathways and ended up in your favorite molecule? And that's particularly important in the context of anabolic biochemistry, where the precursors are generally broken down to generate energy, and then some of the atoms are reincorporated into precursors that end up in proteins, nucleic acids, lipids, complex carbohydrates, and so forth. So the overall approach is that you have your system, your cell culture, your tissue, or your organism, or an organoid, and you provide that with a source metabolite enriched in whichever atoms that you're particularly interested in, and also is readily available, such as carbon-13, nitrogen-15, and then you simply analyze the products at different times after treatment. And we use primarily NMR and mass spec. The main reasons for that is that mass spectrometry measures differences in mass. So if you introduce carbon-13, you increase the mass by 1 atomic mass unit, or nitrogen-15 also increases the mass by 1 atomic mass units. So you can count the number of atoms that have been incorporated into your analyte at different times. And NMR, it works by carbon-13 is NMR active, whereas carbon-12, which accounts for 98.9% of all carbon atoms in biological materials, is NMR inactive. So you're looking at the difference between yes and no pretty well. And similar remarks can be made for other common biological nuclei. Okay, so what you actually do is you determine the specific isotopomer and or isotopolog distributions in the product metabolites. And of course, you also get the total amounts of metabolites. So this means that you get the metabolic steady state, if it is at steady state profile, automatically, as well as information about how many atoms are carbon-13 that derive from your source molecule. And if you're doing isotopomers, which position in the molecule is carbon-13? Is it position 1, or is it position 3, or is it both of them at the same time? Or do you have two molecules in which one is enriched at position 1 and the other is enriched at position 2? They can be distinguished, and I'll show that shortly. Flux analysis takes a fate map that has been developed using the stable isotope traces, and that's the fundamental information and data that's used for determining the rates of the different parts of metabolism, the reactions that occur within the metabolic networks. And that requires a considerable effort computationally. And as I say, that's what I'm going to come back to at the end of the talk. And then you can embed this metabolic information, which is the functional readout of what cells are actually doing, in a systems biochemistry approach, where you combine this information with what you know about transcriptional regulation and proteomic analysis, both of which I should say are fundamentally metabolic, because RNA, DNA, and proteins are all metabolites. They're just big ones. Okay, so this is a representation of a massively simplified representation of a cell. It has an outside, it has an inside, which is predominantly cytoplasm, and then it has internal membrane-bound compartments. And the only one shown with the dashed line represents the mitochondrion. So you have inputs to the cell, so glucose comes in, glutamine comes in, essential amino acids come in. They are taken up, they're transformed metabolically through these networks, and some products are released, such as lactate. If you've got a lot of lactate fermentation, glucose is taken up, and quite a bit of it may be converted to lactate and released into the medium. And then some intermediates are taken up in the mitochondria and further metabolized, possibly coupled to oxidative phosphorylation to generate ATP. And you'll see that some of the mitochondrial metabolites are also cytoplasmic, and there are exchange processes. So citrate that's made inside the mitochondrion can be exported to the cytoplasm, where it's cleaved into oxaloacetate and acetyl-CoA, and that cytoplasmic acetyl-CoA is used for making fatty acids, and it's also the source of acetyl units for acetylation reactions in the cytoplasm and the nucleus, and so forth. Similarly, aspartate is the transamination product of mitochondrial oxaloacetate and also of the cytoplasmic oxaloacetate, and aspartate can come in and out of the mitochondrion, so can malate, and the malate oxaloacetate aspartate cycle with the between the mitochondrion and the cytoplasm is referred to as a shuttle, and that's a means of transferring reducing equivalents from NADH from the cytoplasm to the mitochondrion, because NADH is not directly transported across the mitochondrial membrane. So it's done via reducing oxaloacetate to malate, which goes into the mitochondrion, must reoxidize to oxaloacetate, generating NADH. Actually, that's an oxidation. So the NADH can be reoxidized inside the mitochondrion by respiration and coupled to oxidative phosphorylation. And complex lipid biosynthesis uses dihydroxyacetone phosphate, one of the two 3-carbon sugars from glucose, and that's the precursor to glycerol-free phosphate that goes into the, by condensation with fatty acids, makes glycerophospholipids and triacyl glycerols and so on and so forth. And glycolysis is a multiply branched pathway. In addition to making pyruvate the end product, intermediates go into the pentose phosphate pathway for making ribose for nucleotides, also for making hexoses, for making complex carbohydrates like glycogen, and things like 3-phosphoglycerate is a precursor to serine, which enters the serine-glycine pathway, which is used for making purine nucleobases, and so on and so forth. So sorting all that out with the compartmentation issues, it would be extraordinarily difficult if all you had was the absolute amounts of the various metabolites. One really needs to know where the atoms are going, and that's really the subject of the rest of this presentation. So in terms of cancer models, what you're really interested in is how do you understand the metabolism of a tumor inside a human subject who is unfortunate enough to have a cancer, and so inform on the appropriate treatment modality. Unfortunately, the human subject, which is obviously the gold standard, there are severe limitations on how detailed the experiments can be. So one usually uses simplified models from the simplest two-dimensional cell culture, where you have the maximum experimental control but the minimum relevance, to organoids or spheroids, where you take cells from a human subject and create a 3-dimensional culture, which has more properties in common with the actual tumor. It has a lot of things missing, but it's thought to be a better model, and you can do all of these stabilized state resolved metabolomics on those. And you can go to mouse models, and that has some advantages, and it has a lot of disadvantages as well that I'm not going to go through in detail. I think these slides will be made available to everyone, and this is published, so you can go to the pros and cons of the different models. And we also do human tissue culture with cancer. If the tumor is being resected, we can have access to the actual tumor fresh as it's resected directly in the OR. And the nice thing about that is we also get tissue that is not cancer from the same patient at the same time, from the same organ. So that gives you a paired experimental design, and you can then keep that going in cell culture conditions where you have the control. So, but none of those models on their own is sufficient. One usually has to use a combination of models, and preferably with control over the environment that you can't do with live organisms. Okay, so a very simple example of a metabolic network from the point of view of chemical dynamics. So it's really, really simplified. So if G represents glucose, that can be taken up by the cell and converted to glucose 6-phosphate. That would be the hexokinase reaction. But we're just treating that as one step. And then that molecule has several fates, shown here as going on to another intermediate in glycolysis or going off to substance A, and they are associated with particular rate constants. So the rate of each step would be the rate constant times the concentration of that compound. And then compound X has two fates. It can either go on to make intracellular lactate, or it can go on to make substance C, which could of course be alanine or acetyl-CoA or pyruvate, oxyl acetate. And X could also be made from some other substance that isn't glucose. And then the lactate will be excreted to make extracellular lactate. So if you are only looking at inputs and outputs, treating the inside of the cell as a black box, so you'd look at glucose uptake and lactate excretion, you could set up differential equations of the rate of formation of extracellular lactate, that's the first equation there, is some bunch of rate constants times the concentration of glucose plus some bunch of rate constants times the concentration of substance B, okay? And similarly for the, if you do open up the box, the rate of formation of substance A only in this model only depends on the concentration of glucose and some rate constants, whereas the formation of substance C depends upon the concentration of both glucose and substance B. So I think that you could see that if you were only to measure the amounts of G, A, C, and lactate, you would not be able to determine unambiguously how much of the lactate was produced from glucose and how much is produced from substance B. However, if you were to provide the system with C13 enriched glucose and you measure the lactate, how much C13 is in the lactate and how much C12 is in lactate, you can dissect the pathway into the fraction of the lactate that definitely came from glucose and the fraction that didn't. And then you could try different traces, for example, glutamine, and see how much of the lactate came from glutamine. And if all of it was glutamine, you would identify B as glutamine, or it's not glutamine, it's something else. And in terms of metabolism, of course, none of these, the spontaneous reaction rates are generally very small, which is why you actually have enzymes which are catalysts and speed up the reactions. So in the limit that the enzyme concentrations are low compared with the enzyme concentrations are high compared to substrate concentrations, and or substrate concentrations are low compared with the Km, then all of those reactions are first order in the enzyme concentration. So the individual rate constants can be expressed as the turnover number divided by the Km multiplied by the enzyme concentration. So that gives you a complicated, an additional complication, but it relates the fluxes or the rates to the individual enzyme properties, and that's part of the difficulty of doing flux analysis. Okay. So what I mentioned, isotopomers and isotopologs. I just want to very briefly explain the difference in case there's anyone that doesn't know this. So NMR tends to measure isotopomers, and mass spectrometry tends to measure isotopologs, although it is possible to do isotopomer determination if you have appropriate fragmentation in the mass spectrometer. So isotopomers tell you about the position of the enrichment. So for carbon-13, because you're only concerned with carbon-12 and carbon-13, we can ignore carbon-14 because its abundance is vanishingly low. So for positional enrichment, what position contains carbon-13 versus carbon-12, there are 2 to the power n isotopomers, where n is the number of carbon atoms. And it's the same if it's nitrogen, it would be the 2 to the n nitrogen, or oxygen 18, whatever it is. And you can do something similar if you have multiple labels. So 2 to the power n isotopomers for carbon-13. Isotopologs is the number of atoms. So for carbon-13, where there are n carbon atoms in the molecule, there are n plus 1 isotopologues. So in general, there are fewer isotopologues than there are isotopomers. So to take a concrete example of a 3-carbon molecule, such as lactate, although it could be alanine, anything like, there are 2 to the power 3, which is 8 isotopomers, and 3 plus 1, which is 4 isotopologues. So in mass spectrometry, in principle, you might see 4 versions of lactate. I mean, in practice, you might only see 2, but in theory, there could be 4. There is one in which all 3 carbons are carbon 12, and that would be M0, represented by 3 open circles. There is one way of having lactate in which all 3 carbons simultaneously are carbon 13. That's M plus 3. That has 3 mass units higher than M0. And then there are 3 possible configurations of lactate in which one atom is carbon 13 and the other 2 are carbon 12. And they are indistinguishable without tandem mass spec because they all have identical mass. But they are distinguishable by NMR. They have different, in fact, they have radically different NMR signatures. Similarly, there are 3 possible ways of having 2 carbon 13s and 1 carbon 12. So it's 1, 3, 3, 1, and I think you'll realize that this is the numbers that you get from Pascal's triangle as you go through, as you increase the n. So it's a pretty straightforward thing. So just to summarize again, if you're doing NMR, you measure isotopomers, and there are up to 2 to the n of these things per molecule, and if you're doing mass spec, generally you'll measure the isotopologues, and there are n plus 1. And of course, in practice, so for lactate, if you give uniformly enriched carbon 13 glucose, you typically see 2 forms of lactate. Lactate that has only carbon 12 and lactate that has only carbon 13. There are exceptions to that, depending if you're looking at liver, where you have gluconeogenesis and the scrambling, and then you get some of the other isotopologues formed as well. And of course, observing those isotopologues would immediately alert you to the fact that you have more than just lactic fermentation going on. So it's actually important information. So in terms of NMR, lactate isotopomers, the top shows a region of a proton NMR spectrum that shows the lactate methyl group. So that's the protons of the CH3, and there's also the CH3 of the threonine in there as well. So if we only focus on the CH3, the proton spectrum, then if all carbons are carbon 12, you get the red spectrum that is of those 4 panels, the top left, and that shows 2 peaks because the methyl protons are split by the interaction through the proton-carbon-carbon proton bond into a doublet by the C2H proton. Going down below that, if you have lactate in which there is enrichment at carbon 2 and enrichment at carbon 3, but not in the same molecule, you get that pattern where you have 2 doublets separated by 126 hertz, and that's the interaction of the CH3 protons with the C13 of the methyl. And then there's that group that's near 0 hertz, which is a doublet of doublets, and that's where the CH3 protons are interacting via 2 bonds with the carbon 13 at the 2 position, and by 3 bonds with the proton attached to that. So that gives that particular pattern. If, however, moving to the right, so that's the bottom right-hand corner, you have lactate in which there is carbon 13 at both C2 and C3 in the same molecule, you get a completely different pattern. You get that splitting of 126 hertz and nothing in the middle, but now it looks like a triplet rather than a doublet. So the case where you have a mixture of C2 and C3 versus the case where you have C2 and C3 look completely different. Of course, they would be different by mass spec. In the first case, you have M plus 1. In the second case, you have M plus 2. And in the final case, where all 3 carbons are C13, you get the upper right-hand pattern, which matches the experimental pattern that you see in the NMR spectrum. So that is a 6-line spectra that is characteristic of all 3 carbons in the same molecule. And in the middle, there is some lactate that has no C13 at all. And then next to that is the threonine methyl that shows that there is no C13 whatsoever. And that's a very good sign in mammalian tissue culture, because threonine is an essential amino acid and should not have any C13 present from your source. Okay. So the way we typically represent metabolic networks, you can write down some of these biochemical reactions, the utilization of glucose or amino acids to make pyruvate, which can be oxidatively decarboxylated to make acetyl-CoA and CO2 and NADH for the pyruvate dehydrogenase reaction. It can also be acetate can be attached directly to coenzyme A driven by the hydrolysis of ATP to make acetyl-CoA. Fatty acyl-CoA by beta oxidation produces smaller fatty acyl-CoA and acetyl-CoA, NADH and FADH2, which gives rise to enormous amounts of ATP if the mitochondria are coupled. Acetyl-CoA is also a source of acetyl units for proteins via acetyl transferases, including histone acetyl transferases for epigenetic regulation of gene expression. So if we use this, and many people in the field use this kind of representation of molecules that contain C13, so the 6 dots that represent glucose, that's 13C6 glucose, that's the 6 carbons of glucose are all carbon 13, and they're shown in blue, and that can be phosphorylated to make G6P, and that will go through the oxidative branch of the pentose phosphate pathway to make fully labeled ribose 5-phosphate, and that will enter nucleotides, which then end up in messenger RNA and so forth. The glucose can also go through glycolysis and through the serine pathway end up as enriched glycine, which enters purine at particular positions. Multiple carbons in the purine ring, and that of course is then attached to the ribose that was made through the pentose phosphate pathway. Pyruvate could be fully labeled, and that will make fully labeled lactate, and that has multiple fates. One is through pyruvate dehydrogenase to make doubly labeled acetyl-CoA, or it can be carboxylated by pyruvate carboxylase to make triply labeled oxaloacetate. So these are 2 inputs into the Krebs cycle, and if it goes forward from citrate through isocitrate all the way around to oxaloacetate, that will give rise to, in the first turn of the Krebs cycle, doubly labeled oxaloacetate and the transamination product aspartate, shown by the different representations of oxaloacetate that has 2 open circles and 2 blue circles, and it shows that there's C1 to C4 top to bottom. So because of the succinate to fumarate step, the symmetry of the succinate to fumarate step, and then the subsequent rehydration to make malate, you get 2 forms of oxaloacetate in equal amounts. One is where the oxaloacetate is C13 at the 1 and 2 positions, and the other is oxaloacetate that is C13 at the 3 and 4 positions. They look the same by mass spec, M+2, but by NMR, they look different. And then the transamination product will give you the 2 forms of aspartate, which will end up going into the uracil, which forms the pyrimidine ring of the pyrimidine nucleotides. Okay. Alternative inputs to the Krebs cycle, another anaplerotic reaction, is glutamine. So if you provide the cell with uniformly C13 and N15 glutamine, and here that's represented by 5 red balls for the carbons and two green balls for the 2 nitrogens of glutamine. So the first step might be glutaminolysis. So glutaminase wastes the nitrogen as ammonia to produce glutamate, and the glutamate can be transaminated with oxaloacetate to produce aspartate, and that will transfer nitrogen 15 from glutamate to aspartate, and that is whether the aspartate is labeled or not. So that's catalyzed by GOT1 or 2. The resulting alpha ketoglutarate, of course, is an intermediate in the Krebs cycle. So that would give you, if you had uniformly labeled glutamine, you have uniformly labeled glutamate, and that would end up as uniformly labeled alpha ketoglutarate, and that would end up as uniformly all 4 carbons labeled malate oxaloacetate, which would provide 4 carbons into citrate. So if your source is uniformly labeled glutamine, the citrate after the first cycle would have 4 carbons from the forward Krebs cycle, whereas if it all came from glucose via pyruvate dehydrogenase, it would have 2 carbons, whereas if it was coming from pyruvate carboxylase, it would have 3 carbons, and the positions are also different. So you can see by looking at the isotopomers and isotopolog distributions in intermediates, one can discriminate how the atoms from the precursor molecules go in to make other molecules that are used as precursors for synthesis of macromolecules for biomass. And that is only possible using tracer technology. So that's really where it comes to. So depending on what your question is and how deep you want to go into, you might consider different enriched precursors. So uniformly enriched glucose, where all 6 carbons are C13, is very good because glucose does go into various parts of metabolism, and we use it for survey purposes because it goes into amino acids, nucleotides, lipids via glycolysis, the pentose phosphate in the Krebs cycle, and the hexosamine pathway. So that covers a lot of central metabolism. But if you want to discriminate between the oxidative and non-oxidative branches of the pentose phosphate pathway, that won't do. You'd have to use a different isotoper of glucose, such as glucose in which the carbon 1 and carbon 2 are simultaneously enriched, or you could use carbon 1 and carbon 6. And these are all commercially available. If you're interested in what glutamine is doing, you can use C13, N15 glutamine, or C13 only, or N15 only, and that allows you to probe glutaminolysis, nucleotide biosynthesis, the relative importance of glutamine carbon going into pyrimidines versus glucose carbon going into pyrimidines, energy metabolism through the Krebs cycle, and fatty acid biosynthesis, reductive carboxylation, and so forth. C13 enriched fatty acids for beta oxidation and fatty acid biosynthesis, C13 serine for serine metabolism, 1 carbon metabolism, and of course the lipids that contain serine, the ceramides and sphingolipids and so forth. And C13 glycerol is important in lipid biosynthesis, among other things. So one might use many of these in a very deep study, or if it's more targeted for very specific questions, you would choose your source accordingly. So looking at isotopomers by NMR, I've already shown you something about that can be done by one-dimensional NMR. Sometimes you have to go to two-dimensional NMR, and the technique has the horrible acronym total correlation spectroscopy, and that only looks at through bond interactions, which means that you can do all this in mixtures because everything is intramolecular. So the bottom line is, if it's carbon 12, you have a simple spectrum. If it's carbon 13, you have a more complicated spectrum. And depending on the actual distribution of the isotopomers, where the carbon 13 is, you get completely different patterns. Even if they're pure isotopomers or mixture of isotopomers, they all give rise to distinguishable patterns, which furthermore can be quantified, so you know the relative abundances of each of the isotopomers. And this is this kind of thing that you get in practice. On the left is where the source molecule is uniformly enriched glutamine, and on the right, the source molecule is uniformly C13-rich glucose. So in glutamine, this is just focusing on two molecules in part of the spectrum. At the bottom left is aspartate, looking at the carbon 2, carbon 3, the protons attached to that. And at the upper right part of that panel is the glutamate, and actually the glutamate in reduced glutathione, showing the interaction of the 2 carbon with the 4 carbon and the 2 carbon with the 3 carbon of glutamate. So as you can see in the picture there, the 1 position is the carbonyl at the top, and the 5th position is the carboxylate at the bottom. So what you see with the glutamine is that the aspartate, there is a square array with a blob in the middle. The blob in the middle is where the aspartate contains carbon 12 at both the C2 and the C3. The actual square, shown in the green box, the four vertices of the square represent the aspartate in which both the carbon 2 and the carbon 3 are simultaneously C13. There is the case where you have 2 peaks that show the molecule in which the carbon 2 is C13, but the carbon 3 is C12. And there is the case where the carbon 2 is C12 and the carbon 3 is C13. So there are 1, 2, 3, 4 different quantifiable isotopomers determinable by this experiment. And the quantitative analysis gives us a great deal of information about the input to the Krebs cycle, the number of turns, and the output, which in this term is the precursor to uracil. So if we look at the aspartate entering the uracil, immediately below that, the glutamine, you see at the top right there is uridine monodiene triphosphate, and we're looking at the 5 and 6 positions of the uracil ring of UTP. Okay, so we have exactly the same pattern that you get with aspartate, because aspartate is an immediate precursor. And the 2 and the 3 position are what give rise to the 5 and 6 position of uracil. So this is telling us about pyrimidine biosynthesis in these cells. And you get the same kind of pattern with the CTP next to it, and CTP is produced from, well, UMP. So if you look at a different carbon source, so this shows that glutamine is a very good carbon source for making aspartate and pyrimidine nucleotides. If you look at glucose as the source, where the system has unlabeled glutamine, but now has C13-rich glucose, you do see very similar patterns, but the relative intensities are considerably low. There is much more of certain isotopomers, particularly the unlabeled ones, in glucose than there are for glutamine. So if we go through the quantitative analysis of all these isotopomers, we can determine the relative importance of glucose versus glutamine carbon input to the pyrimidine rings. And in this case, about 2/3 of the carbon came from glucose and 1/3 came from 2/3 came from glutamine and 2/3 came from glucose. Just to show that NMR can compete extremely well with immunology in terms of incomprehensible acronyms, I'm just going to show you some of the awful nomenclature that we are guilty of in this field. So there's glutamate again, showing the structure, and this would be glutamate that is enriched in carbon 13 in all 5 positions plus nitrogen 15. So different NMR experiments can decipher different labeling patterns of the molecule. So the TOXI gives us information about the C2, C3, and C4. This thing called HACACO, which correlates the H of the C alpha, C2, with the C alpha, C2 itself, with the carbonyl of the 1 carboxylate. and HACN does the nitrogen, carbon, hydrogen, and so on and so forth. And you can use these experiments to edit a very complex spectrum and pull out only those molecules that have specific patterns of enrichment. So if you look at panel D on the right, which is an experiment, that will display only those molecules in which there is carbon-13 with a proton attached to it that is also attached to a carbon-13 with a proton attached. So you can see that lactate and allene are indeed enriched in that manner, as indeed is glutamate and glutamine, as indeed the ribose subunits of the free nucleotides. They're fully enriched. And that's how you tell in detail for proof what isotopomers you have. Okay, and just to show that one isn't restricted exclusively to carbon, you can do the same kind of experiment detecting the nitrogen through the protons. Here there's 2 of the ring nitrogens in this is AMP, detecting through the protons the N3 and the N9, which come from the amido nitrogen of glutamine, and then the other cross peak is the N1 comes from the amino nitrogen of aspartate. So in this experiment, because it must be N15, and we provided N15 enriched glutamine, we know that that aspartate amino group must have come by transamination of oxalacetate with glutamate derived from glutamine. So we know that GOT1 and or 2 are active in these conditions. So a little bit about mass spectrometry. It pains me greatly to have to say that mass spectrometry has some value. If you use very high resolution mass spectrometry, you can tell the difference between a molecule that has 1 nitrogen 15 versus one that has 1 carbon 13, even though they have the same nominal mass, 1 AMU higher than the unlabeled molecule. And that is because the neutron in nitrogen doesn't weigh exactly the same as the neutron in carbon 13. When you add a neutron to nitrogen 14, you add 1 AMU almost, and when you add a neutron to carbon 13, you add not exactly 1 AMU. And with high resolution NMR, it's actually quite trivial to resolve those differences in masses, as this shows. So these are baseline resolved peaks for nitrogen versus carbon enriched glutamate at a resolution of the order 400,000. So what that means is that you can do multiplexed mass spectrometry where you can supply the system with both carbon 13 and nitrogen 15 at the same experiment. And because of the mass resolution, you can determine whether what you're looking at has how many carbons and how many nitrogens. And that's quite an advantage. One can look at macromolecular turnover, proteins, DNA, nucleic acids, complex lipids, complex carbohydrates. They're all made from small metabolites, amino acids, acetyl-CoA, S-adenosylmethionine, monosaccharides, and they're often modified. So you can measure the precursor biochemistry by the small molecule metabolomics, but the macromolecule product is also accessible either directly by lipid analysis or after hydrolyzing the big guys to their constituent small molecule like amino acids, nucleotides, or carbohydrates. So there's a literature on intact lipids, protein hydrolysis. We have a new method of rapid hydrolysis of proteins that doesn't destroy the amino acids like the conventional method does. And we can measure the stable isotope distribution of the resulting amino acids with high accuracy. With RNA and DNA, you can enzymatically hydrolyze to the nucleotides and then measure them by mass spec or NMR. And similarly, by complex carbohydrates, you can hydrolyze them down to glucose or whatever the other carbohydrates are. So I'm running out of time, so I'm going to go quite quickly now through some examples. lipid biosynthesis. This is a breast cancer cell line, extraordinarily slow growing. And we're just looking at the global lipid synthesis. So that's the total neutral lipid and phospholipid content of cells after being exposed to different C13 carbon sources. So C13 glucose, you get this, and we see the C13 is entering the glycerol backbone of the complex lipids, and also the fatty acyl chain. So both parts of the lipids are being synthesized as the cells divide. If we use glutamine in the presence of high glucose and under normal levels of oxygen, we see with these cells no detectable incorporation of glutamine into either the fatty acids or into the glycerol. Whereas if we use glycerol itself, we don't see incorporation into the fatty acids, but you see it unsurprisingly into the glycerol backbone. Whereas if we use octanoate, a medium chain fatty acid, which is water soluble, goes into the cells easily. Unsurprisingly, it doesn't go into the glycerol. It's not a acetyl-CoA is not a glucogenic precursor, but of course it enters the fatty acyl chains. And the details of the pattern there shows that the fatty acyl chains, the acetyl-CoA incorporation, 2 carbons at a time, requires that the acetyl-CoA pool comprises doubly labeled acetyl-CoA and unlabeled acetyl-CoA, and the acetyl-CoA is being used for resynthesis. So it's not only that the octanoate is being broken down, it's being used for resynthesis at the same time. So that seems to be something that cancer cells do that adipose or liver cells tend not to do. They're either making it or not making it. Okay, I'm going to skip the next one and give you an example of a hexosamine pathway. So hexoses are used for making complex carbohydrates, and they're also used for adding to proteins for glycosylation. The hexoses themselves are typically not terribly reactive, or will react indiscriminately with reactive groups on the proteins or the lipids or carbohydrates. So this is controlled by first activating the hexose to make a hexose sugar, probably the commonest of which is UDP hexose, like UDP glucose, precursor for making glycogen, UDP hexosamine, which is used for transferring the hexosamine subunit to proteins for O-link glycosylation, N-link glycosylation, and so forth. So the hexosamine pathway is quite interesting because it involves numerous pathways that all act in parallel and come together to make the product that is used for transferring the hexose component. So we have the structure of UDP. This is UDP and acetyl glucosamine. So U for uracil, R for the ribose, that's the UDP part, G for the hexose, which is the origin is glucose, and A for the acetyl. So we have glucosamine, we've added nitrogen to the C2 position of the glucose, and then acetylated. So if you supply the cells with C13 glucose, you have glycolysis that produces pyruvate, produces acetyl-CoA, ends up as C2 oxaloacetate, goes into aspartate, and then that goes into the uracil, 2 carbons of the uracil component of the UDP gluconate. The C13 glucose also goes through the pentose phosphate pathway, and that ends up in the ribose component of the UDP, and that adds 5 carbons to the ribose. Glycolysis coming out as fructose 6-phosphate. That is where the addition of the nitrogen occurs using an aminotransferase with it's actually an amido transferase. It uses the amido nitrogen of glutamine to produce glucosamine 6-phosphate, and that's enriched at all 6 carbons, and then it's acetylated, and the acetyl group may be unenriched, depending on whether it came from glucose or from other source, or it's doubly labeled if it came from the glucose. And that makes the acetylglucosamine, and then that is isomerized to make N-acetylglucosamine 1-phosphate, and is condensed with UTP to make UDP N-acetylglucosamine. Okay, so it's a complicated and interesting system. So if you use a combination FTMS and nuclear magnetic resonance, on the right is the time course of the deconvoluted system. So we're considering the UDP glucnac as these 4 metabolic subunits: uracil, ribose, hexose or glucose, and acetyl. So the acetyl can exist either as unlabeled or both carbons are C13. There's 2 forms of the acetyl. The glucose can be either completely unlabeled or all carbon 13, no other isotopomers. The ribose can be either unlabeled or has all 5 carbons, and the uracil can be unlabeled, 1 carbon, 2 carbon, or 3 carbons. So the total number of species to consider is, in terms of isotope logs, is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 nominally. But of course, the sum of the glucose is equal to 1. There is 16% unlabeled, 84% labeled. So the 2 are not independent. So the number of independent is 10 minus 1, 2, 3. There's 7 independent parameters to optimize, and there are 17 isotopologs to measure. So it's an overdetermined problem. And that was deconvoluted into these metabolic subunits as a function of time. And that shows how the data and the estimates agree for the different isotopologs. And if you look at the actual deconvoluted time course, the square open squares are the M0 unlabeled. So that starts at some value and decreases essentially exponentially as the originally present unlabeled UDP glucnac is used up, and its total concentration is maintained by synthesis of new material. You see that the open circles is the M+5. That is the ribosyl part, and that reaches a steady state value of 90-something%. And actually the rate constant that describes that exponential process matches the rate constant for the decay of the unlabeled species. And then if you look at the filled squares, that is the uniformly labeled 6 species, and that shows a lag because here it goes through fructose 6-phosphate, and then you add the amino group, and then you add the acetyl group. So that's contingent on earlier steps in the reaction, which shows a lag. And similarly for the uracil and the acetate. And interestingly, the uracil never reaches anything like above 90% enrichment. And that's because, as we said earlier, 2/3 of the carbon, thereabouts, doesn't come from glucose, it comes from glutamine. So you wouldn't expect the uracil enrichment to reach more than 40%, say, total. And similarly, the acetyl group also does not plateau out at steady state at high levels. It's somewhere of the order 30 to 40%, which means that there is another source of acetyl groups other than glycolysis and pyruvate dehydrogenase, which in fact is numerically more important than the glucose source. We actually are not entirely sure what that is in this system yet, and that requires additional tracer studies to work that out. The other thing to notice is that the time taken to reach isotopic steady state, even for this particular metabolite, is more than 24 hours. So if your flux analysis requires that you're at isotopic steady state, you really have to push the system for a long time. And I think that I don't have time to go through the remainder. Sorry for speaking too long, so I'll just conclude that the stabilized state resolved metabolomics is the enabling technology to permit atomic tracing in metabolic networks inside cells, inside tissue, inside whole organisms. As you increase the complexity, the amount of effort required to get unambiguous information increases, but it is, at least in principle, possible. It is also possible to multiplex different stable isotopes by NMR and ultra-high resolution mass spectrometry. Metabolic networks by these means can be uncovered in, as I said, cell culture, tissue and organs, and even whole organisms, mice and human subjects. Flux modeling can be carried out once you have the fate maps and sufficient temporarily resolved metabolic data. So it does require a lot of experimental work. And it can be integrated with transcriptional and protein level information in what might be loosely called systems biochemistry. And that provides ultimately mechanism, how things are happening inside tissue, inside the organism, which is very useful in preclinical drug development, analysis of response, in monitoring, and indeed even in screening, so that you're really looking at the organ of interest for your treatment. Is your tumor really resolving because it's doing what you think the drug is actually doing? And just finally, I want to acknowledge my co-directors of the Center for Environmental Systems Biochemistry, Teresa Fan, Richard Higashi, and Hunter Mosley. And we have been generously supported by NIHS, the National Cancer Institute, and NIDDK. Thank you.
Well, thank you very much for really a wonderful talk, Dr. Lane. I certainly would think it's fair to say that this is a very fascinating topic, and it's certainly very complex, and it does require, I think, quite a bit of biochemical knowledge. But again, really a nice overview of this sort of technology and this sort of approach. So we do have a couple questions that we will go through. These are in no particular order, but certainly we would like to hear your feedback. So one of them you kind of touched on yourself a little bit towards the end, but there's certainly quite a bit that can be done with this sort of approach. And one of the topics that's, I think, an integral aspect of that is the difference between achieving steady state equilibrium, particularly as it relates to isotopic equilibrium, versus maybe doing more of a kinetic analysis. And in that context, perhaps you can comment on the two, and perhaps how long does it actually take to achieve what is otherwise isotopic equilibrium, so that you get a full view of all the potential distributions of the atoms, so to speak.
Yeah, that is a difficult question. So if you're doing cell culture, central metabolism will reach isotopic steady state quite quickly, and within probably a couple of hours, or for some metabolites, even within minutes. If you put in glucose into cells, glucose gets taken up very readily, and it's converted very rapidly through glycolysis. So, and the content of the glycolytic enzymes is pretty high. So glycolysis will tend to equilibrate quite quickly, provided your control over the cell growth is well controlled. So the other problem that you have is the internal rate of metabolism fast compared with the production rate of things like biomass. So in the microbial field, the way that people typically do this is in a chemostat, and there you can pretty well guarantee isotopic and metabolic steady state by working with a chemostat. There's not so much work done under chemostat conditions with mammalian cells, partly because of traditions of cell culture, and partly because they're much more fragile than microbial cells are, which have a tough coat. But progress is being made, particularly with microfluidics, where you can capture cells in microfluidic devices and flow through. And there you can achieve isotopic steady state, certainly in terms of inputs and outputs and central metabolism of the cells themselves. So it depends a bit on what it is you're looking at. Lipid turnover and protein turnover are much slower than, so anabolism is almost by definition much slower than catabolism. So the catabolic processes tend to equilibrate relatively quickly, but some of the anabolic processes can take quite a long time. So we did recently publish a paper doing this kind of work with mice. So what we had done in the past with mice was bolus injection of the tracer into the tail vein. And so the advantage of that is that you go straight into the bloodstream and it distributes through everywhere within a minute. It's very, very fast. And the metabolic rate of mice is phenomenally high. I mean, it's, you know, the normalized rate compared with the human, it's 7 times as fast as a human. So the time scale is very much compressed for mice compared with humans. And, you know, if you go from a mouse model to a human model, you really have to take that into account, the differences in intrinsic metabolic rates. So if you do a bolus injection, of course, the glucose concentration in the tissue starts high and then rapidly drops as insulin goes up, and then the C13 enrichment in the glucose drops quasi exponentially over a period of an hour or so. And then if you look at the C13 enrichment in the lactate in the peripheral blood, that starts off at 0, goes up to a maximum, and then comes down again, and reaches a maximum, at least in the strains of mice that we were working with, 15-20 minutes. So 15 minutes is a good time to sample the tissue. However, because you've done a bolus injection, you're not going to be at isotopic steady state. This is a pulsed method, so it's intrinsically transient. So you cannot apply steady state computational methods to that kind of thing. You have to solve the time-dependent differential equations and get out the transient response. Now, people who have a background in enzyme kinetics know that there is always more information in a transient experiment than there is a steady state experiment, at the cost of considerably more effort in the computational analysis. But the steady state analysis has more ambiguities than the transient analysis does. So ideally, you'd like to be able to do, you have the choice of doing a transient experiment and one where you at least approach isotopic steady state. So for isotopic steady state, you've either got to do continuous infusion, and there are groups that have been doing that. There's a Yale group that's been doing it for brain studies. There's the group at UT Southwestern that have been doing it in mice and also humans, where you first give a bolus of C13 glucose and you follow up with a continuous infusion over a period of roughly 3 hours. And then if you look at the tissue concentrations of C13 metabolites, the central metabolites have more or less plateaued at about typically 3 hours. I mean, it will vary a bit from tissue to tissue because of uptake rates and so on and so forth. But they're not truly at isotopic steady state. So the question is, okay, if you're 90% of steady state, is that an issue? Probably not. But if you're only 50% towards steady state, you can't really use the metabolic flux analysis methods. You have to use the fuller approach, which is a lot harder. But there are groups that have developed software for doing exactly that. So we did, and so that's only 3 hours, and quite a lot of the slowly turning over pools of macromolecules do not reach isotopic steady state. So if you're interested in the slowly turning over pools, you have to extend that labeling period. Well, you're not going to do that with humans. You're not going to infuse humans for 24 hours. You're probably not going to infuse mice for 24 hours with C13 enriched glutamine either. It'll cost you too much money. So we developed a liquid diet so the mice eat when they want, and the diet contains the sucrose that's normally, the carbohydrate that's present in the normal chow is replaced with C13 glucose, and it's liquid, so they slurp it up. And they like it, and they eat at lib, so they come out at night and they eat. So you probably have typically 8 hours of eating, and then when you sacrifice them, you look at the stable isotope enrichment, and we've done that for different periods. So after 12 hours, the majority of the larger metabolites have reached isotopic steady state, but not all of them. Some of them are still increasing. So the answer is yes, and it's no, and it depends. So one has to do some preliminary experiments on the system with respect to the question that's being asked before splurging on doing the whole analysis.
Well, thank you for that answer, and I think that is very, very insightful. So for sake of time, we'll just have two more related questions. But one with respect to SIRM, obviously you mentioned NMR quite a bit, and mass spec. So obviously with mass spec, you have both LC and GC-based approaches. So I was wondering if you could comment on GCMS-based metabolomics, and also how does SIRM compare to some of the other analysis softwares that are available, such as isotopic ratio outlier analysis?
Oh, okay. So GC is nice because the resolution is fabulous. GC is horrible because you have to derivatize to make things volatile, and it usually means that the number of standards that you have that you're running, interleave mode, your coverage is moderate. Many mass specs that are coupled to GC are low resolution, not much better than unit mass. So for SIRM, you can't do multiple labeling because you can't tell the difference between carbon 13 and nitrogen 15 simultaneously. And there are different derivatization methods. They all have their pros and cons, but GCMS, it's pretty good. It's very good for amino acids, for example, and it's pretty good for hexoses if you use the right derivatizing. I mean, the resolution is really quite extraordinary. If you can distinguish epimers of the hexoses by GCMS, I think that's pretty impressive, because most LCMS simply can't do it. There are so many versions of LCMS, and whether you go to ultra fast LCMS or you want to use the slower ones that have different properties, so reverse phase and Hillick, and we use some LCMS. We use ion chromatography LCMS. Its advantage is that it's fantastically good for phosphorylated compounds, whereas GCMS is completely useless for that. And most LCMS just doesn't resolve most of the phosphorylated intermediates that are present in the pentose phosphate pathway. ICMS, we get them all. So that's really valuable. And we get all of the negatively charged species. It's lousy for amino acids for reasons that we don't fully understand. So you have to use multiple platforms. And that increases your overall time. If you have to use 3 mass spec platforms, that's a lot of time and resources, and potentially a lot of sample that you need. We also do direct infusion mass spec, so which is the fastest you can possibly do, at the cost of you can't possibly get any information about isomers unless you're extremely lucky with the tandem, where you have different fragmentation patterns for structural isomers, which occurs, but of course that doesn't discriminate enantiomers. In fact, resolution of enantiomers is just an extremely difficult problem by any analytical technique. NMR doesn't do it either. there are some techniques, but they're very hard to apply to DNL enantiomers, and the same is true for mass spectrometry. It's very difficult. You tend to have to be lucky with your column. So IROA is primarily an identification technique. So you know, you get the fully labeled and the unlabeled isotopes, and because it has a predictable mass difference, you use that to help you identify those metabolites that actually can be made from a C13 precursor. So that involves a selection, and in mammalian systems, of course, that means you don't get the essential amino acids. Whereas if you're doing untargeted methods, even if valine, leucine, isoleucine, threonine, and phenylalanine are not C13 enriched, you're still measuring them. If you use an untargeted method, you measure everything, and you also measure all of their isotopologues and isotopomers. You can't get isotopomers from IROA. You don't really get the isotopolog distribution, the quantitative distribution from IROA. You have to do, you have to go and measure and resolve all the individual isotopomers and or isotopologues and determine them quantitatively. So the purpose of IROA is different from the purpose of SIRM. But being able to identify molecules unambiguously is very important, because mass spec is actually not terribly good at identification. It's also not terribly good at quantification. You absolutely have to have an authentic standard, because if you only look at ion intensities, you can be quite wrong about the amounts. And I just wanted to put in about what we all measure by NMR and mass spec, and pretty well any analytical technique. We do not measure concentration, ever. We measure the number of moles by NMR, the number of moles of material, or in mass spec, you remember the number of moles of ions. And they have to be back calculated to the number of moles of the particular analyte in your original sample. And to declare a concentration, you have to know what the volume was. I mean, that's easier for biofluids. You usually know what the volume of blood was that you extracted. But what you don't necessarily know is how many of those metabolites were bound to proteins that were precipitated when you did your extraction. So you don't know exactly what it is you measured. Now, if you do NMR of raw plasma or raw urine that's had no extraction or derivatization, what you will measure is the amount of the free metabolite and not the bound metabolite. And in principle, although no one ever actually does this in practice, you make that measurement and then you precipitate the protein, sorry, then you denature the protein and precipitate it, and then remeasure it, and the amount there is the total. So that allows you to discriminate between the free and the bound. And biochemically, free and bound compounds are quite different. This becomes a major issue for the metabolic flux analysis of cells, where you have multi-compartments and multiple pool sizes that are a priori not known, and B, quite difficult to determine with any reliability, even in cells in culture. So that is another problem that one has to face, and right now do the best you can.
Wonderful. Well, thank you again very much for your insight, and really a wonderful talk, Dr. Lane. It is definitely appreciated. So for sake of time, again, we're going to go ahead and finish up. So again, I do want to thank all attendees for this webinar, and certainly, again, a very big thank you to you, Dr. Lane, for giving a really fascinating and wonderful talk. I do want to remind everyone that we will be having upcoming webinars. At the moment, we are going to try to have one planned for January, but at the moment it is not scheduled. So please stay tuned, and we will release that information once we get it available. But again, thank you everyone for attending today. And again, thank you very much, Dr. Lang, for really a wonderful talk.
Thank you. Appreciate it. Have a good day, everyone.